Welcome back, everybody, and it gives me great pleasure to be chairing for the Emerging Scientist Award. So it's an award we introduced at DDL to really to fill a gap that we had. So we've long had the Pat Burnell Young Investigator competition, which we'll be running this afternoon, and we always start with the DDL lecture. But we thought we're not really doing anything for people in the earlier stages of their career. And so we created this award to recognise people who were making a significant impact on inhaled drug delivery. And in the competition, this year, as every year, we had excellent applications. There's a panel of people that judge the award, and what we're looking for is somebody within 15 years of their PhD, and then we look at the applications, which are the winners' applications going back, the people that you see on the screen here, or all on the DDL website. You can see the kind of quality that people are submitting, and we evaluate those for the relevance. So are people addressing an unmet need in inhalation science? For the innovation, the novelty and creativity of the work which people are reporting to us, the scientific quality and the rigor, the cogency of the application, the sort of narrative of how people have developed their careers, and then their impact, their influence that people are having on the field of drug delivery to the lungs. And so this year, as I say, we've been through the process again, and we have a winner which we announce at this stage in the conference. So can we have the next slide, please? So I'm very pleased to say that this year's Emerging Scientist Award goes to Catherine Froman. Catherine's got an engineering background, which she then is applying to inhaled drug delivery, and have a particular interest in mucosal immunology. So we'll invite Catherine to the stage now to come and give us the Emerging Scientist Award lecture and tell us all about what she's been doing.
The plaque. Oh, thank you so much. Thank you. Oh, thank you so much. First, I have so many thanks to give to start this. So thank you first to the judging committee for recognizing the impact of the work. Thank you all for being here, coming back from the break before lunch. And thank you to DDL for just being a really fantastic community to be a part of. It's really, looking at the people who've come before me with this award, they're inspirations to me. And so it's just really an amazing opportunity to be counted among these fantastic investigators. And I hope I do myself justice with my talk today and live up to the great standards that those before me have set. The other thanks I need to give is to my research group. And so I had the fortune to speak to the new research investigators last night. And I told them that now as an academic, my product is really my students, right? And so I have a fantastic group of engineers, biologists, chemists at the University of Delaware that are really the ones who are leading the work. And so everything that I'm going to talk about today is really theirs, and I just get to get out of their way and try to inspire them to solve important problems. And so hopefully what I talk about today will really build on the themes that we've already heard for this conference so far, thinking about yesterday, talking about models for drug delivery and advancing our decision-making preclinically, as well as thinking about innovation and translation and how we get things out of the academic lab with fantastic students and their creativity, and thinking about how we influence the field. So I have a personal anecdote about this. I first came to DDL back in the 2010s as a graduate student myself. And so it was, I did my training at NC State in chemical engineering in a drug delivery lab that was really had a platform for creating nanoparticles. And my PI said, okay, well, put these in the lung. That's easy, right? And I took myself to DDL. I thought this was a community that I could come and learn. And what I found very quickly is, as we all know, it's very challenging, and that's not such a trivial thing to do. But I was able to immerse myself in aspects of the lung and the lung biology and the barriers to drug delivery. And I became fascinated with the immune system. And when we think about inhaled particulates coming from that nanomedicine background, they end up, and every textbook says, right, they end up in some sort of phagocytic cell. Alveolar macrophages are the cellular clearance mechanism, right, that stops all of your drugs from going to their actual targets. And so these aren't great targets, but they're that important clearance mechanism. But I became really fascinated with this because these immune cells are really the conductors, the orchestra conductors that are in the lung mucosa that are regulating homeostasis, right? They're clearing debris, but they're also sending signals to all sorts of other cells, be them barrier, stromal, or other immune cells. They're able to migrate, they're able to launch adaptive immune responses, and so they're really powerful in directing immune responses. And with an inhaled particulate, you have an immediate way to interact with them. And so these cells obviously are super important to maintaining health, but as your lung changes, whether it's healthy aging, whether it's a disease state, right, the microenvironment that these cells find them in really changes. And so these cells can be still conductors of good, but they may have to change their tune, their pattern, their responses. They may become exhausted. They may be actually the rogue conductor that's contributing or causing the disease. And so I hope that I won't go into too much immunology here because I don't think that's the take home, but I want everyone to feel accessible that these cells are really instrumental in regulating the immune environment and thinking about them as an opportunity for us as we are developing new inhaled products. So about these cells, these antigen-presenting cells, I'm thinking about macrophages, I'm thinking about dendritic cells. There's a number of different specialized populations in the lung, but they're really capable of doing some conserved functions. So the first and most important one that we think about is phagocytosis, particle clearance, right? They're able to engulf actively and digest different sorts of debris or our inhaled therapeutics. They're also able to present antigen, and so they're able to sample the environment, digest it, present these onto different billboards that then signal to other cells, either through direct contact or some soluble factors. And so their ability to undergo antigen presentation, travel to lymph nodes, leads to adaptive immune responses, and then these become localized immune responses within the airspace. So you can have mucosal-specific antibodies, mucosal-specific effector cells that help, again, regulate the immune environment. These cells, as you can imagine, are highly specific to that immune environment. So whether it's inhaled particulate that they're getting signals from, or their local cell-cell contacts, ECM interactions, soluble factors, and they can take on different phenotypes. And so we'll largely lump them into pro or anti-inflammatory types of states where they're able to then do different functions. So they become responsive to lots of things, which as engineers means there's lots of knobs that we can tune to potentially modulate them. But these are also the things that cause disease and contribute to this. So if they go into a rogue state for too long, that's often exacerbating disease. And as we look at every pulmonary disease, we can usually find dysfunction in the antigen-presenting cells and these phagocytic cells that are contributing to an exacerbating disease. So to convince you that they're good phagocytes, I'm showing you on the left, these are 3-micron particles. When they turn pink, they're inside the cell. And so these cells are able to roam around, engulf foreign particles, start to digest them, and they also can digest other dead cells in this video as well. And because of this responsivity to different environmental cues, right, they're able to execute a complex playbook. They can do a lot more than we can do with a single small molecule because they can do things temporally, sequentially, and they can really engage a whole different range of activities than just a single input itself. But we, as the engineers, have that input of the particle. And so from a very simple input-output, if you're able to give the right cues and instructions to these immune cells, you can think about getting them to do something desirable and getting them to change the local immune environment. High level, correct a bad response or create a good response. So prophylactic vaccination, right, engendering local protection in the lung, or thinking about the disease state and the immune dysfunction and seeing if you can correct it. So sounds easy. Of course not, never is, right? as you think about the lung, depending on where you're at within the lung architecture, the composition of these immune cells changes. And because we're not able to get in there and look and track it every single cell and understand how they're responding in every single scenario, there's a lot that we don't know about the different populations of antigen-presenting cells and how they're responding in different locations. But certainly the barrier properties change, the abundance of antigen-presenting cells change and are different locations within the airspace. So again, if you're trying to access these as the modulators of the immune environment, you have to know where your therapeutic is getting to know which cells you're actually engaging with. So this is all the background for questions and things we think about in my lab. And so when you look at my portfolio, it almost feels like jack of all trades. We look at intracellular signaling up to deposition modeling across these multi-length scales. And this is where the engineering training, I hope, comes into play. Thinking about chemical engineers from down the molecular level, what's happening with inside a cell, cell-cell signaling, and then up to the tissue level architecture. All to really understand how we can better understand and predict immune responses to inhaled products. And so this is a universal tool set, things that I'll talk about, certainly as we're thinking about different formulations and drug products, but really our goal has been to harness the information we get to correct and drive specific immune responses. So my lab has really been focused on developing tools at all these different length scales to understand immune responses and ultimately direct them for different disease states. And so it's a universal approach. We're not necessarily disease specific. Hopefully you'll see how our different approaches can play into different pulmonary diseases. So really focus on inhaled. Okay, so in these engineering tools, we try to think a little differently of what's missing in the field and what is the knowledge that we don't have at all these different length scales. And so for the rest of the talk, I'm just gonna give little snippets of things that we're working on currently that I'm most excited about, but we have really played in all of these different length scales. So really trying to highlight things that are missing within different length scales. We'll start with the smallest, start in the intracellular and move up to the kind of tissue level at the end. So this seems like maybe a simple question, but we asked this when I started my career of what happens when that innate immune cell internalizes a particle. We all know that these macrophages, dendritic cells, they undergo phagocytosis, clearance, the end, right? But that's not the end, right? These cells are so integrated in all these other processes that there has to be more than that. And so how do we really understand and fill in this missing gap of what happens after this clearance in how these cells are regulating their microenvironment? And so to answer this question, we've developed a model platform particle that we use for lots of things. And we like it for a number of different reasons. Mostly it's very modular. We are able to control all sorts of physiochemical properties of it. And so this is based on a PEG diacrylate hydrogel system. We make it through a reverse emulsion polymerization where we can tune the monomer composition to present different surface charges, different degradability, cross-linkers, and control the size as well. So generally we're working in 300 to 500 nanometer sized particles. Our base formulation is slowly degrading, and we kind of like that because it allows us to track things over time. But we can certainly change the crosslink density and composition in order to drive degradation. We also can put fluorophores in here that are covalently attached, so again, we can track the particles over time. Now, my title slide maybe offended people. I said it's immunologically inert, and you're gonna say, PEG, there's somebody in there who always thinks PEG is not immunologically inert. I hear you. In our hands, we're looking at these for acute studies, and this is whole cell proteomic data, heat map. And so hopefully you can see right here, I have a nanoparticle treated group, an untreated macrophage group, and an LPS, or a bacterial wall component treated group. There's really stark differences with the LPS in terms of inflammatory response, and we're comparing the nanoparticle to the untreated group. There's really minimal changes that are happening. And so while we can argue about long-term adaptive responses of PEG, in the single cell first interaction with this, there's really minimal effects that the cell is experiencing. So this allows us to isolate the question, how does internalization alone of this physical thing, without any inflammatory effect, without any other stimulatory cues, really drive responses in the cell? And so it's with this model system, we've done a lot of different things, understanding degradation profiles and cellular components. We've done this in vivo as well, looking in murine models, and I'd like to show some relatively recent, kind of surprising results that have led us on a path of understanding this process even further. We went ahead and we instilled an intratracheal installation of these PEG diacrylate nanoparticles. We put them into the airspace of a mouse, and we see rapid uptake in alveolar macrophages. By 24 hours, almost all of the macrophages have taken up our particles. And then we did a lavage, and we just looked at the concentration of extracellular vesicles. And so hopefully you can see from our mock PBS control compared to the nanoparticle group, we see a really increased difference of extracellular vesicles. That was kind of surprising to us because across the board, there's no inflammatory cytokines. There's really no other changes. There's no, you do histology, you're not seeing anything. It's a 24-hour time point, it's really acute. Now, we can't say that these are specifically from macrophages, but we knew that this was kind of the end clearance state. These are specifically small EVs, so the EV nomenclature is, for someone getting into it, is a big pain. But basically, depending on their biogenesis route and their size, you can sort of classify these, and so we'll call them small EVs because they're sub 200 nanometers, and their biogenesis route is likely through these intracellular processing mechanisms. So we don't know that these are from macrophages, so we just grew some macrophages in a dish, we dosed them with different amounts of particles, and we see a really strong correlation between the number of particles you give a cell and the amount of EVs it puts out. That was kind of surprising to us, because again, these cells are not exhibiting any other inflammatory profiles. Is it this magical PEG diacrylate nanoparticles or something about it, the charge or whatever? And the short answer is no. So if I take my untreated cells, there's a baseline amount of EVs it secretes. If I put even a cationic or an anionic particle, we see an elevation, so it doesn't care about charge. If I take a polystyrene sphere that's 100 nanometers, so much smaller, we don't see the effect. But if I take a 1 micron polystyrene and I give it to the particles at the same mass amount, I do see the effect, so it is size dependent. And if I stimulate the cells with LPS, I don't see the same increase. So this was actually really surprising to us because the conventional thought is you stimulated a cell, you activated it, it's probably changing its intracellular processing, metabolism, and therefore the EVs will go up. But it really seems to be linked to this active process of phagocytosis. So we've done a lot of work on the signaling. I don't want to necessarily go into it, but I'd like to show you some nice pictures of cells because pictures are great. So these are cells that have received our particles. We can zoom into different sections. So if we look on the right side here, we can see the lysosome is this dark region. There's these white patches. Those are our particles that are hydrogel things that are slowly degrading over that 24-hour time period. And next to every lysosome we find are these multivesicle bodies, or MVBs. And so multivesicle bodies are known to be part of EV biogenesis, but they've never really been linked up to lysosomal degradation and specifically phagocytosis. And yet we see them physically co-localated within the cell, and they increase in abundance the more particles we're giving. So this is kind of our working mechanistic picture where we're really finding links in these red arrows that we've indicated here that are, if you look at any review paper that looks at phagocytosis or EV biogenesis, these are big question marks. And so we don't have all the pieces yet, but our evidence is really saying that there's something specific about phagocytosis. And again, our macrophages in the lung being big phagocytosing cells, and how they're then regulating EVs and that EV cell signaling. Why should we care, right? This is inhaled drug delivery, yes? So if you give these cells some particles, they are going to start to digest that particle, and they're packaging that material into EVs. So we've looked at the EVs themselves. We can start to see the same fluorescent dye that we gave in the particles over a lag starts to show up in the EVs. And that actually can be transferred to other cells. So here I'm showing some naive macrophages that never saw a particle, never saw that fluorescent particle. But hopefully you can see on the projector here a little Cy5. That's that red signal that came from the particle. So this is EVs that came from cells that received nanoparticles that are now getting that transferred. It's not localized into endosomes. It's now in the cytosol, and so it's a different mechanism of internalization. And so as we think about inhaled drug that goes to a macrophage, depending on the life cycle of the molecule, the particle, these things are going to be packaged into extracellular vesicles, and those signals are now being spread out to other cells and processed in through different mechanisms. And so this is a way to amplify signals. These EVs have antigen presentation capabilities, transferring those sorts of things. And so we think this is a lot of opportunity to engineer the not only intracellular response, but think about how it propagates signals to other cells in that microenvironment. So that brings me to my next transition to think about how do we think about the microenvironment and what are other cues that those innate immune cells are responding to. And so that's literally the question we've asked, and how do we build platforms that allow us to really study and understand how those different extracellular microenvironment cues are affecting immune cell phenotype. And so this is in collaboration with my colleague April Coxon at the University of Delaware. We've had a number of co-advised students that have worked on this problem. And when we think of the gap that this is really trying to fill, I think many of us, myself included, have always been guilty of doing everything in tissue culture plastic. So these rigid substrates that are in the gigapascals, and we culture our cells and we ask, do assays of toxicity or, immune response. That's very different than the microenvironment a cell is going to feel. And so lung tissue is on the order of maybe 1 to 20 kilopascal. So orders of magnitude difference in stiffness. And it turns out the cells care, right? And so we've been working to really develop a platform to be able to modulate this in a very narrow way that can better replicate aspects of disease. So we think about healthy lung tissue, and as that stiffens and the extracellular matrix around it is stiffening over time, as say in something like fibrosis, we see maybe from 1 to 20 kilopascal change. Really subtle in the scheme of moduli, but we ask really, does that stiffness and the presentation of those cues start to affect the cells? So how do we go about this? We make a synthetic extracellular matrix. And we come back to PEG, seems to be a theme in my career. It's slightly different chemistry. It's a 4-arm PEG where we can put different linker peptides and pendant peptides that represent components of the ECM that we can control entirely synthetically. So if we want to have more laminin presenting or fibronectin presenting signals, we can really start to tune those to better mimic in a controlled manner the extracellular matrix environment. We do a free radical polymerization under aqueous conditions, and we make a soft hydrogel where, as we tune the weight percent of our monomer components, we can really achieve these very narrow moduli ranges within the microenvironment. This has also been scaled to a commercial platform, so now we can start doing this through ink dropjet printing and put cells in there as well. So we can do this at 96 well plate and high throughput sorts of assays. So I said it matters, so here's some little evidence to support that. We can do lots of things with these hydrogels, so we can put cells on top of them. And so on the left, I'm showing a soft gel, on the right, a stiff gel. There's also a cytokine that's present, but hopefully you see a lot of green in the stiff, but not in the soft. The green here is a marker of that kind of wound healing M2-like phenotype that a macrophage might take on, and it's overabundant in fibrosis. And so if we then do a phagocytic assay of these cells on these different microenvironments, we see in fact that these cells in that M2 wound healing phenotype are less effective at taking up a particulate. And so right off the bat, you can see that these subtle changes in stiffness alone, that's the only thing that's different about the gel, is really instrumental in changing how an immune cell is responding. You can also think then about embedding the cells within this gel. So maybe you're thinking about interstitial macrophages, dendritic cells, incorporating these stromal cells as well. And here we're looking at the same kind of soft-stiff comparison, the same kind of moduli range with P. aeruginosa and looking at bacterial uptake. And again, we see that same phenotype replicated where there's actually a decrease in phagocytosis in that stiffer microenvironment. So this is, ongoing work. We're building these gels, and we have the ability to tune lots of properties about it. We're also able to work with any number of other cells. So here we're looking at epithelial cells that we can culture on top of the gel. So now they see a unique basement membrane of the right moduli. But because we have synthetic control over the gel, we can start to do things with the chemistry where we can basically replicate a scratch assay, but using light. And so we can selectively degrade aspects of the gel, remove parts of the barrier, and now study a wound healing response in situ. And so we can see how these barrier cells are able to form and heal across different scratches, what matrix they're depositing, and as we incorporate other cells in the gel, see how that cell-cell signaling is really aiding or hindering the response depending on the microenvironment. And so there's a lot of opportunities with this to think about systematically building in complexity as you need. We talked a lot about, you have to find the right model to the right question. But these are really now enabling in a high throughput manner to do our traditional 96 well plates in submersive culture, ALI culture, with controlled not only ECM components, you can really tune in which are the ECM signals that are driving responses, but also which cells and in which abundance are really there. And so we have this platform now to augment a lot of the other in vitro assays that are there to now incorporate aspects of the ECM microenvironment in a really highly tunable, customizable way. So it gets me through the microenvironment piece, and so we're going to go out to the tissue level. And when I started this way back many, many years ago, I was always struck by not having a really great predictive experimental tool that shows you where things deposit in the lung. And we've talked yesterday certainly about the importance of breathing profile. Just because you have the right size particle doesn't mean it goes where you think it does. And certainly as you overlay disease and ventilation on top of that, knowing and predicting where things deposit is forever our challenge. And so we leaned into this question to see if we could develop an experimental tool that would allow us to look at an entire inhaled dose on a realistic breathing profile and provide us that complete spatial deposition. So maybe we're crazy, but here's what we've done so far on it. So we can take a CT scan, upper airways, out to a few generations, but certainly that's not encompassing the entire surface area and volume of the lung. So we can't manufacture this at the scale, at the complete resolution we would want to make a lung. We don't want to do that. We just really want to be able to discretize it spatially. And so we said, let's start with the easiest unit. Let's start at the lobe level. Let's compartmentalize these and see if we can design something that would allow us to build in the right surface area, the right approximate right flow profiles in order to collect particles in a spatial sort of manner. So we called this tidal, which maybe was not a useful name because you want a tidal model to do tidal breathing, but anyhow, we thought we were clever at the time. This is the model we came up with. We take the CT scan, we truncate it at the main bronchi, so you can take any organic upper airways. We have an idealized mouthpiece, but this is easily interchangeable. And then we made these lobe units. These lobe units are representative of the human air lobes. And then within each of them, they are sealed. So we have silicone molds at the end that allow us to actuate them and move in and out to create realistic breathing profiles. And they're populated with lattice filters on the inside that allow us to collect, spatially collect our aerosol. And so really these are now enabling that spatial collection, but also to be able to impose a whole range of breathing dynamics. And certainly it's all 3D printed in some capacity, so it's highly modular and tunable. So we actually went and built it. This is what it looks like. It sits on a lab bench, the top in our office in Delaware. And so you can see the 3D printed airspaces, the different lobe units. There's independent motors that are connecting each of these lobes. We have a flow meter that's measuring out the mouth, and so we can do kind of your normal spirometry-based breathing profiles. And we can impose those breathing profiles by tuning the motor and the motor positions. If I zoom into one of those lobe units, these are the filter designs that we came up with, and we sort of had to plant a flag in the sand because you could do an infinite amount of things and you want to get to what's good enough, but also not over-defined. And so we have effectively two regions. We call them central and peripheral. They are the average hydraulic diameter of a certain range of generations within the airspace. It's tunable. We can swap out the unit cells, the porosity, the flow through this. But really, we were inspired by thinking of two regions, central peripheral, so that we could ultimately be able to benchmark it to scintigraphy data. Now, scintigraphy being 2D, our model is 3D, and so we have to do a planar transformation in order to account for potential overlap depending on the configuration. But we essentially are able to have these 3D printed lattices collect aerosols. Just like an NGI, we would rinse them, take it apart, rinse them, and then we can build up three-dimensional localization for our model. So this is out of the lab at a little demo we did. There's a little piece of paper in front of the mouth so you can prove that it's breathing. For how much work it is, it's maybe not the most exciting video, but proof it does breathe. And we really have a range of breathing features and parameters. So we can obviously tune the rate, the duration, the extent. We can get volumes from 500, 700 mils up to 1.5 liters. We can do this fast, slow, peak. We're starting to change the shape. And all of these are actually moved symmetrically, but there's no reason that we can't offset them. And so we have all the controls set to do this, to be essentially mimic what would be a realistic profile, whereas more lower lobed breathing as opposed to upper airways, and all of these things are tunable. The other thing we can do is put breath holds in there. So it's not shown, but it's trivial to just pause the motors for a little bit, right, and put a breath hold. So now you can start to get and impose these really realistic breathing profiles onto this model. And so just one set of data, heat maps of the sorts of things we can generate. We took a fluorescent molecule and an aeroneb, 2 different size aeroneb nebulizers, expose this for 5 minutes, rinse it, collect it, put spatial heat maps. These are replicates that we've then repeated. Important for us, and it took a long time, we can completely close the mass balance, so we're getting 90% or more recovery of everything we put in comes out. And we have a washing procedure so that we can really recover and get to baseline amounts so that we're able to reuse the parts. Certainly we can print them again, but you can imagine the amount of printing that goes into this is non-trivial. And so we're able to get spatial deposition maps. We've done a number of ways to benchmark it, and everyone who does deposition modeling, what is a good benchmark? So we've looked at scintigraphy, published results for these same breathing profiles. We've done MPBD benchmarks, and we're within that, kind of 15-20% mark across the board of where we end up. So we're really expanding this profile with things that I'm not sharing here, but hopefully you can appreciate smaller particle got a little lowered lung deposition, less upper airway, less mouth throat from our larger aerosols. And so where do we see this going? Certainly we can control all sorts of aspects of these filters. And so our ability to impose regional obstructions or deteriorations to mimic aspects of lost surface area, and having representative patient cases for how the internal structure changes. So it's interstitial lung disease in certain regions that we could actually map back to a patient CT scan. I'm not suggesting we should personalize this for everyone, but if we had some benchmark cases in that preclinical development stage where, hey, your therapeutic, your device is targeted at severe COPD, let's run it through this model to make sure your formulations are getting to where it needs to go at the right amount, and how do off-target effects and representative breathing profiles start to affect this. You can also then think about tuning the ventilation through the motor control to, again, have mimicking not only what the capabilities of a patient who has lung disease. And so it's altered ventilations at the lobe level, but also the extent, duration of all the breathing capabilities that they might have. And so if I put this all together, we really think we have positioned this to be an additive value to the field, where we have this deposition model that now can have internal structures and breathing profiles that mimic aspects of disease. But then what this really does that I think is a critical missing part of all of our new emerging ALI in vitro models is to dose them correctly. And so now you can think about using the information from the spatial deposition model to put the right amount of your therapeutic onto these culture models that then can be tuned to mimic aspects of disease. And so having representative, a series of representative deposition models, but also then a series of representative culture models that look at upper airway, middle airway, lower airway in different disease states, and then be able to dose them correctly. And so we're really trying to build towards that integrated pipeline, not putting cells in our plastic model, knowing it's a plastic model has its own limitations, right? But that these become complementary tools that allow us to understand more completely about specifically immune cell and immune responses within the lung in response to inhaled therapeutics. So that's really where our lab has been. We have, again, hopefully shared with you today some different tools and different approaches, trying to fill in missing gaps about how we predict immune responses in the lung at that cellular microenvironment and tissue level scale in order, again, to be predictive and hopefully provide some additive utility in ultimately driving immune responses and thinking about immune engineering, but in the short term also being able to provide a surrogate for immune responses and outcomes. And we're really looking to partner and translate this. We're a small lab, but I think there's a lot of opportunity, and we're really positioned now to start trying these in many situations. So if this is of interest to you, I'd be happy to talk about this after and thinking about how we might potentially work together and how our tools can better solve problems that you're having. So with that, I'd like to thank my lab, my funding, the judges and organizers again for really this fantastic honor. Again, just super humbled to be here and really appreciative. And I'd like to thank you all and happy to chat with you after this session. Thanks.